Hi friends, so let us continue with the next video, and that is pharmacokinetics. Under the pharmacokinetics, we have already discussed in the last video the four important points, that is absorption, distribution, metabolism, excretion. These are the basic for the pharmacokinetics. And now in this session I'll be discussing about the point that is absorption. Okay, so what absorption, what does it tell actually? If you observe carefully here, let me read the points written here, that is absorption, right? You have something known as intestinal lumen here. The moment you take the drug, it goes into the intestine. So this is an intestinal lumen, and this intestinal lumen, right? So if you observe, so example, if this is the stomach, okay, this is stomach, okay. So these are the things we need to understand, okay. So this is the drug, and this drug is absorbed into the blood stream, okay, in the bloodstream. Now the drug, it is taken into the bloodstream. How this drug is absorbed in the blood vessel lumen. So the first thing written here is, you have something known as lipid, refers to biological membranes. So these are the biological membranes. That means now this is what? This is the GIT. Okay, say example, this is an intestine. Okay, this is the blood vessel. Now I have taken the section of this. Okay, so you got a lumen here. You can see this is lipid. It's nothing but it is a biological membrane. That means it is the lipid cell. This is a cell. Okay, so you got a like cell membrane, all that. Okay, now if you observe carefully here, the subcutaneous insulin gets filtered into the capillary through aqueous channel. And if you observe carefully here, you have a drug, few drugs which have a channel which passes through this channel, enters into the blood vessel lumen. And such examples are subcutaneous insulin, which gets filtered into the capillaries through an aqueous channel. This is an aqueous channel, right? Next point written here is, you have something known as passive or simple diffusion happens across a semi-permeable membrane. If you look into the points here, you don't have any channels which helps in the transport of a drug from the lumen into the blood vessel. This start diffusing. Okay, now if you observe carefully here, it only requires the concentration gradient. The moment you take the drug inside, the concentration of the drug in the GIT is increased. The moment the concentration of the drug increases in the lumen, so it moves from higher concentration to lower concentration. And this movement of higher concentration to the lower concentration through a semi-permeable membrane, right? Through a semi-permeable membrane, this is known as passive or simple diffusion, right? So let us look into the next important aspects. The next important points written here is, okay, if you observe carefully here, you have something known as carrier mediated transport. So observe carefully here. Okay, you have something known as carrier mediated transport. What is written here? It's written that receptors or pumps present in the lipid layer are called carriers. Okay, drugs bind to the carrier, takes the drug into the circulation. Now in the same scenario, this is a lumen, this is the blood vessel, and you can see these are the lipids. That means this is a cell. Now in this type of carrier mediated transport, the name itself is telling carrier, right? It has something like receptors or the pumps present in the lipid layer, right? What does it do? They start, okay, this drug bind to the carrier, and take the drug into the circulation, right? It take the drug into the circulation. Now second point we need to understand, right? There is a carrier 1 and you got a carrier 2. Carrier 2 also you can see the same arrow mark is different. But what is the difference between the carrier 1 and the carrier 2? Okay, so carrier 1 tells it is a facilitated diffusion or passive diffusion happens with concentration gradient. So if you observe carefully here, this is a facilitated diffusion. Okay, this is a facilitated diffusion. Now this is an active transport. Now what is the difference between the carrier 1 and carrier 2? The only difference between the facilitated diffusion and passive diffusion is the involvement of the carrier. Now he has compared, this is a facilitated diffusion. Okay, this also we spoke in this chapter in the previous topics, previous sentence. It's written that this is a passive diffusion. Now what is the difference between the passive diffusion and the facilitated diffusion? Is asking important question, and the answer is carrier, right? Only difference between the facilitated diffusion and the passive diffusion is a carrier. Now if you observe carefully here, you have something same, facilitated diffusion or the passive diffusion. This happens with the concentration gradient. Now it needs a higher concentration. If the diffusion happens from higher concentration to the lower concentration with the help of a receptor or the pump, this is known as, what is that? This is known as facilitated diffusion or passive diffusion. Now what actually happens in the active transport? Okay, now this active transport, okay, in active transport, what happens in the active transport? It moves from lower concentration to the higher concentration. D is a lesser concentration. Now the drug which is present here is at the lesser concentration compared to the substance which is present at the D point, that is higher concentration. To move from lower concentration or the lesser concentration, lower concentration or the lesser concentration to the higher concentration, it needs an ATP, right? It needs an ATP. So we call that as active transport. Okay, we call that as active transport. Now what are the examples of this active transport? We'll be discussing in detail in this video itself. The example is P-glycoprotein pump. Okay, you have something known as P-glycoprotein pump. We'll be discussing that. Okay, now the next important point written here is for absorption of glucose. Okay, if you look into the carefully, for the absorption of glucose, you have something known as GLUT receptors. Okay, GLUT receptors are facilitated receptors, and SGLT1 and SGLT2 receptors are active transporters. Whenever glucose is consumed, SGLT in intestine absorbs glucose against the concentration gradient. Okay, in SGLT2 in kidney reabsorbs glucose against the concentration gradient. That means these SGLT1 and 2, they are active transporters. Okay, now say example, if this is the intestine and you have the SGLT1, this SGLT1, right, the concentration here is low and here it is high. As we discussed here, it has to move from higher concentration to lower concentration. But SGLT, it is what? It is an active transport. So the absorption of the glucose from the intestine into the blood vessel from the lower concentration to the higher concentration, this is an example of active transport. Same in the case SGLT2. Say example, if this is the kidney, in SGLT2, the moment the glucose is present in the urine, in the tubules, these are the cells absorbs, okay, absorbs the glucose against the concentration. So that is the reason our glucose level is maintained and there is no production, or it is not eliminating the sugar or the glucose out. Next we have something known as GLUT4 in skeletal muscles. It allows glucose uptake depending on upon the concentration gradient, right? So we need to understand, okay, what happens in the GLUT4. GLUT4, it is all about the, you can see it is a facilitated diffusion. GLUT4 is facilitated diffusion. Now the moment the GLUT, so this is the skeletal muscle, this is the GLUT4, and this is the level of a glucose. Observe carefully, this is the level of the glucose. The skeletal muscle only absorbs from the higher concentration to the lower concentration, not like a GLUT1 and GLUT2 receptors which are present, okay, GLUT1 and GLUT2 receptors which are present in the where, in the intestine or in the kidney from the respective aspects, okay. So they are not like that. So that is the reason this GLUT4 plays an important role. It only absorbs only when the concentration is high. Now the other important points we need to understand here is most common mechanism of drug absorption, passive or simple diffusion, right? The most common mechanism of drug absorption is passive or simple diffusion. In this simple diffusion or passive diffusion, there is no receptors as compared to the facilitated diffusion where you have a receptors, okay. The next important aspect is drug given by oral route enters the intestinal lumen, right? Intestinal lumen. The moment it is distributed to tissues along concentration gradient by passive diffusion, right? P glycoprotein moves the drug out against the concentration gradient. This is drug efflux, right? So let us understand what is it trying to tell us. Now what I do is, this implies that drug is eliminated before it reaches the systemic circulation, right? PGP causes first pass metabolism of pre-systemic elimination. The first pass metabolism is a misnomer, right? It is seen in liver and intestine. Now what is it trying to tell us is, the moment we take the drug, okay, the drug enters into the, this is a lumen, okay, this is a lumen. Now you have a drug here, you took the drug, okay, it is here. Now the drug, what is it? This is the intestinal epithelial cells, okay. Now you still no need to confuse, you got an, okay, you have an intestine here, so you got a, now what is this? This is a cells. Now I've taken one section of the cell here, right, and I drew it here. This is beautiful. So you took the drug inside. Now the drug enters into the these cells, epithelial cells, right? Normally it absorbs into the bloodstream. Now what happens here is, now the moment you take the blood, it enters into the intestine, it is absorbed into the blood, distributed to tissues along concentration gradient by passive diffusion. So because it has gotten higher concentration here, so it is absorbed. Now you have some kind of receptors known as P glycoprotein pump. Now what happens, you have a receptors which is present, if you observe carefully. Now the drug which is present in these epithelial cells, right, they are kicked out into the lumen, back into the lumen by this P glycoprotein pump. This kicking out the drug through this P-glycoprotein pump is known as drug efflux, because it is moving the drug outside efflux. So this is a drug efflux, right? So this implies the drug is eliminated before it reaches the systemic circulation. Before it reaches the circulation, the systemic circulation, it is eliminating out from this systemic circulation. So it is known as pre-systemic elimination, or this is a first pass metabolism, right? And remember that first pass metabolism is totally different. It is a misnomer. And this type of the drug efflux, right, it seen in the liver and the intestine, right? So that's all you need to understand. The next important point here is there is something known as pathological application of active transporters. And what do you mean by pathological application of active transporters? Now say example, you have an cancer cells or bacteria. Drug enters the cancer cells or bacteria. The moment the drug enters the cancer cells and the bacteria only based on the concentration gradient, but cancer cells or bacteria move the drug back to the lumen using active transporters against the concentration gradient. Active transporters in bacteria or cancer cells act as mechanism for the resistance, right? So if you observe carefully here, now what is this? The pathological application of active transport, right? What is a pathological application? Now what happens in the cancer cells of bacteria? Now the drug enters into the cancer cell. Say example, this is the cancer cell, right, or the bacteria cell. Now but the cancer cells or the bacteria cell, right? Now since the concentration is high in the blood, it moves from higher concentration to the lower concentration. Now the lower concentration is cancer cell or bacteria. But what happens here is this bacteria or the cancer cells, they move the drug back into the lumen. So what is this? This is an active transport. Okay, they move the drug back into the lumen using an active transport against the concentration gradient, right? This active transport in bacteria or cancer cells act as mechanism of resistance. That's the reason the moment you give the drug, it is taking inside, but it is not acting in the cell. It is kicked out of the cell through the active transport, right? Through the active transport. So that's all you need to understand in this session. Let's meet in the next session. Thank you.